Hi everyone, today's lesson is on mitosis and meiosis. Mitosis is general cell division for eukaryotic organisms. This is what most of our cells will do if they're going to divide. Now for cell division, we know that as cells grow, that it's hard for them to get the nutrients in and the waste products out if they get too big. Remember our surface area to volume ratios? So before this happens, we divide the cell into smaller ones, and we use a process called mitosis. Mitosis is what is done by eukaryotic cells only. This is because we are looking at dividing up the nucleus. Prokaryotic cells don't have a nucleus. In mitosis, we are making a copy and then separating the cells. Now, making copies of our chromosomes, it's important to know what a chromosome is. So we start with DNA. We wrap that around histone proteins, kind of like wrapping string around a spool. And then we keep twisting and twisting and twisting and twisting, and it gets thicker and thicker and thicker. Imagine taking your shoelace and twisting that shoelace until it starts winding up upon itself. We've all tried that. So that's what happens here. And it condenses, and as it condenses tighter and tighter and tighter, we wind up with our chromosome. Now here we have one chromosome, and this is the copied part. We call these sister chromatids. Our cell cycle is a lot like a clock. Okay? And you'll see that in this clock here, interphase takes up most of this circle, with it having three smaller phases. Then you see mitosis and cytokinesis. Interphase is about 90% of a cell's life cycle, and it is divided into three different stages. First, we have G1. G1 or gap 1 is where we increase in size or grow and synthesize new proteins and organelles. So this is after we've divided, we have to go back to our normal daily activities. At that point, we might have the cell triggered to go into a replication or division, mitosis, and we'll enter our S phase. In the S phase, our DNA synthesis takes place. So we make the copy of the DNA. So the copying of DNA happens during interphase. This isn't actually part of cell division. Next, we go into the G2 phase, and this is where it prepares to divide. That means we have to increase the amount of cytoplasm and organelles within the cytoplasm. So here we see cells in interphase. Not very distinct. After interphase, we enter mitosis. This is the actual division of the cell, and it is divided into stages as well. It has 4 different parts to it. We have prophase. In prophase, the chromatin condenses into our chromosomes. Everything is packed up and our nuclear envelope disappears. If it's an animal cell, we'll see the spindles forming and the centrioles just moving to the ends of the cells. Next, we have metaphase. During metaphase, all of our chromosomes are going to line up along the equator, right along the middle. Metaphase is in the middle. This is so I can make sure that it's organized and I can move one copy to each side. You'll see that it looks a little bit different in plants versus animals. Plants tend to have much bigger, thicker, longer chromosomes than animals do. Next, we enter anaphase. In anaphase, we pull those copies apart. So that centromere, the part that was holding them together, is broken, and we separate them. You can see here in the animal and plant cells, it kind of looks like teeth on a monster. Next, we go into telophase. In telophase, we've moved everything to the 2 poles. We're going to reform that nuclear envelope. You can see in our animals and our plants there. And then next we have cytokinesis. In cytokinesis, we're going to, for animals, basically pinch it in half. We have this cleavage furrow where it starts to push in, and it begins to squeeze around the middle and eventually pop it into 2. Imagine taking your belt and making it so tight you popped it in half. It's kind of what happens there. Now in plant cells, it can't be the same because they have the cell wall around it. So what they do instead is they have to make new cell wall in here. That happens by the Golgi body making vesicles that contain the material for the new cell wall. And they all move to the center there, and then they pop together. If you've ever done dishes and play with the bubbles, you can kind of pop them together, make bigger bubbles. That's what happens here. So it pops together and makes this big, long string that eventually winds up becoming all the way across and all the way up and down, making our new cell wall. A shorthand way of remembering mitosis. Okay, here's a mnemonic for you. I picked my apples today. I for interphase, that's in between. P for prophase, where we pack up our DNA. M for metaphase, where it's all in the middle. A for anaphase, where we pull them apart. T for telophase, where we get two nuclei. Then don't forget cytokinesis, where our cytoplasm splits. For those of you who are visual learners, here's a concept map. We can divide it into two different parts. We have our cell cycle, which is divided into interphase and mitosis. In interphase, we have G1, where is our normal growth and daily activities, S, where we synthesize our DNA, and G2, where we get ready for division. We have to make more cytoplasm and organelles. Then we enter mitosis. In mitosis, we divide it into prophase, metaphase, anaphase, and telophase. In prophase, we pack up our DNA, we get rid of that nuclear envelope. In metaphase, we line up in the middle. In anaphase, we break the centromeres and pull them apart. And in telophase, we make two new nuclei. And then cytokinesis, where we split them in the whole cell in half. There is a short video on mitosis that's by Crash Course. These are amazing videos. If you ever get a chance, go watch the Crash Course videos. They have them on everything. Okay, so I'm going to take a minute to show you this video. Okay. So I've explained the different steps. You watched a video on the steps, and I hope Hank was able to explain it to you well. There's a lot going on, but there's just some basic steps to remember. Now here you can see, this is a slide of an onion root tip. So this is plant cells, and you can tell based on those boxes around them, those are their cell walls. And this slide shows the different stages of mitosis happening. Now, the roots of the onion are going to be growing very rapidly because they're moving down into the soil. So you can see here that we have a lot of cells that are going to be in interphase. You can't really tell what's going on there, but you can see the dark on the inside. Those are the nucleoli. So the nucleoluses that make our ribosomes. Then you can see where they start to pack up. Here we go. There's one. That one's in prophase. Metaphase, where they line up. And just happens to be right next to it, we have anaphase, where they're pulling apart. Now I don't really see, oh wait, there we go. There's a telophase here in this slide. You can see that cell wall being formed in the middle there. Very cool. Now if we look at, these are animal cells. You can see different stages here in our animal cells. Most of them are going to be pretty nondescript with just little dots inside. Those are all in interphase. Remember, it makes sense that we would see a lot of them in interphase. After all, they spend most of their life in interphase. After that, we have prophase. And in prophase, we're going to see it all starting to pack up. Okay, so we would say that this one here is in prophase. Here we have one in metaphase, anaphase. And on this slide, I never did find one in telophase. Okay, so that is mitosis. Make sure you send me an email, talk to me during office hours. Let me know if you have questions so I can help you out with mitosis. Check out part 2 so you can see my